So let's go ahead and put a lot of things together. You know about enzymes, you know about carbohydrates, you know about coenzymes from the previous video. You know that ATP is an important molecule and a molecule that holds a lot of energy and is a useful molecule for a cell to have. Let's put all those concepts together and talk about certain aspects of cellular metabolism. Specifically, what we're going to describe here is carbohydrate metabolism, and even more specifically, glucose metabolism. Glucose, of course, is a monosaccharide. We covered that back when we did carbohydrates a number of weeks ago. So cellular metabolism covers an awful lot more ground than what we're going to do. You can talk about protein metabolism and lipid metabolism and so on and so on. But we're zooming in on just some specific aspects of cellular metabolism because we want to answer the question, where does a cell get its ATP from? Or at least where does it get most of its ATP from? We talked about how important ATP is and how useful it is, active transport and lots of other things that a cell does with ATP. Well, where's all this ATP coming from? And one of the possible answers to that is a cell can take a carbohydrate like glucose, extract energy from that carbohydrate, and use that energy to build ATP. Use it to attach phosphate groups to ADPs, and that's how you make ATPs. So how does that happen? Well, we're going to answer that question in really a three-step process, or three-phase process. So we're going to talk about the catabolism. Remember, catabolic means breakdown as opposed to anabolic. So the breakdown of carbohydrates, specifically glucose, and we're going to break it down into three sets of reactions. And if you flip forward in the lecture outline book, you'll see that I've used the same notation there with the Roman numerals. So the first set of chemical reactions that we need to investigate is called glycolysis. Glyco, as you already know, means carbohydrate. It refers to a carbohydrate. Well, we're going to be using glucose, which is a carbohydrate, to start this whole process, so that makes sense. Lysis is a root that means to break something down. When you lyse something, you cut it apart. So glycolysis literally means cutting apart a carbohydrate. So we're going to watch this series of chemical reactions in which we start with glucose, chop it up, and remove parts from it. And as we do so, we're extracting energy from it. And we're going to use that energy to build ATP. But we're going to go further than that. We're going to take the end product of glycolysis and chop it up even more in a series of reactions called the Krebs cycle. And then we're going to take some of the products of the Krebs cycle and use them to jumpstart, if you will, this process called the electron transport chain, another series of chemical reactions. So each of these is a series or a set of chemical reactions that happen in different places and are distinct from each other, but are certainly related to each other. And if we think of them as, phase 1, phase 2, phase 3, we've got a pretty good picture of the way most cells deal with their carbohydrates, specifically the way most cells are going to metabolize their glucose. There are exceptions, but most cells do these processes. Certainly animal cells, like human cells, do these processes very efficiently. So let's start off with a discussion of phase 1. That's glycolysis. As usual, top of our page, we've got some blank spaces here. So as you run across terms that you might not be familiar with, fill them in here. Be familiar with the terms that are here. And let's look at glycolysis. Glycolysis is a catabolic process. In other words, we're going to break something down. Specifically, we're going to break down glucose. And as we do the steps of glycolysis, we're going to oxidize the glucose, meaning we're going to remove electron equivalents from it. Review the video on redox. We're going to make ATP. We're going to use the energy from glucose to drive the production of ATP. That's a good thing because cells love ATP and it's always useful to have. And we're going to steal some hydrogens from glucose and give them to NAD molecules, which means we will have produced NADH molecules. As you will see coming up in the next couple of videos, that's a valuable molecule. I've, in the past, used an analogy. I said ATP is sort of like money. It's like cash, right? If I have ATP and I'm a cell, it's like I have $100 bills in my pocket. Well, NADH is also valuable, but it's like having, say, a credit card. It's not cash, but you can still get something done with it. It still has potential value. It still has a value to you. Even though it's not cash, it's spending power. So both this molecule and this molecule are useful. We're going to make both of them in this process called glycolysis. And where's the energy coming from to make this and this? It's coming from your original starting material, glucose, the energy-rich carbohydrate. So it would make sense that if we're going to use glucose as our starting material, that we should become a little familiar with it. You already know some things about it. If you go back to the carbohydrate section of the lecture outline book, we've drawn glucose before. In fact, we drew one of its isomers, fructose, as well, right next to it on one of those early pages in the book. So let's take a closer look at it again. It has the chemical formula C6H12O6. By the way, I've drawn it as a straight chain of carbon atoms with hydrogens and oxygens attached. You may see it in other references drawn as a ring of carbon atoms. They're both acceptable ways to draw glucose. Anyway, so there's the formula. This is an incredibly energy-rich molecule. It's a very highly reduced molecule. It has a lot of reducing power. It just means it has a lot of electron equivalents. That's still another way of saying it has a lot of hydrogen atoms. Remember from the previous video, I'm using that to represent a hydrogen atom. So of the 24 atoms it contains, 12 of them, half of them, are hydrogens. That's an incredibly energy-rich, highly reduced, valuable molecule. And it turns out it's pretty easy to get. It's the most common monosaccharide in nature, so it's readily available. So if a cell acquires this stuff, it says, great, I got a great fuel molecule. I got a lot of potential energy here. If I can figure out a way to extract energy from this molecule, I can use that energy to build ATP, which is really what my goal is here. So if you were to take a look at the whole process of glycolysis, you would see that it's really 10 chemical reactions, one after the next after the next. So here's your glucose. There's an enzyme that uses it as a substrate and converts it to this other carbohydrate. And then another enzyme converts it to this. And then another enzyme converts this to this, and another to, and so on and so on. So you have this whole series of 10 chemical reactions until you wind up with this carbohydrate at the end called pyruvate. So do we want to delve into each reaction in depth? Well, that's an important thing to do, but not for right now. What I would like to do is introduce you to glycolysis, talk about where it happens, why it happens, what you start with, what you end with, what's the purpose of doing it, and then it's one of those topics that comes up over and over and over again in biology. So you'll study glycolysis from a slightly different perspective in, say, a physiology course, or a microbiology course, or a cell biology course. It keeps coming up for lots of reasons, and each time you see it, you'll understand it more and more and more deeply. If you take a biochemistry course, you'll understand the chemistry of each of these reactions very deeply. I think for our purposes in this course, it's most important to say things like, well, why do you do glycolysis? Where does it happen? What do you start with? What do you get out of it? And how does it relate to those other processes? So that's what we're going to do. Now that said, I would like to spend some time focusing in on just the first of these 10 reactions for several reasons that you will see. So we're going to go into this first reaction in some detail. We're not going to do that level of detail for all the other 9, but let's spend a few minutes talking about the first step of glycolysis. I'm going to write this a little bit bigger so that you can see it better. In the first step, the first chemical reaction of glycolysis, we're going to take, as we said before, a glucose molecule. There's a glucose molecule. And we're going to take a closer look at it, first of all. So let's say I'm a cell. I just acquired a glucose molecule. I'm all excited because I have this energy-rich molecule. Great, let's metabolize this thing. Let's get some energy out of it. Let's use that energy for something productive like ATP production. Well, that's all well and good, except it turns out that glucose molecules aren't terribly reactive. So if I want to perform some chemical reactions on that thing, I got to get it to want to participate. I have to get it to be interested in participating in chemical reactions. So it's very rich in energy right now, but not a terribly exciting molecule, not a terribly reactive molecule. I got to make it more reactive before I can expect it to participate in the chemical games that I want it to play. So one of the common ways to get molecules to be more reactive and to be more exciting and to participate in chemistry, you see this all the time in biochemistry, a common way to do that is to add a phosphate group to it. Remember phosphate groups? We talked about them back when we did phospholipids. We said they have these negative charges on them. There's a negative charge, there's a negative charge. And it turns out when you add a phosphate group to an otherwise boring, not so reactive molecule, you make it much more reactive and much more chemically exciting and much more participatory in chemical reactions. So the first step of glycolysis is going to be sort of a setup to do future steps. You're going to kind of wake up the glucose molecule so that it wants to participate in chemistry. Specifically, you're going to add a phosphate group to a glucose molecule. So let's see how that looks. To make that happen, we're going to need an enzyme that catalyzes this reaction. I'll name that enzyme in just a minute. But that enzyme will pick up a glucose molecule and we'll find a phosphate group, more on that in just a second, and it will release as a product the following molecule. What I'm drawing here in blue is exactly the same. It's identical to what's over here in the glucose molecule, in the previous molecule. In other words, so far, I'm drawing what looks like an identical molecule, but as I proceed, I'm going to change one thing. Down at the 6th carbon, we're going to make a change. What if at this 6th carbon, 1, 2, 3, 4, 5, 6, at this 6th carbon, instead of having 2 hydrogens and a hydroxyl group, like I do on glucose, instead I have 2 hydrogens, just the oxygen part of the hydroxyl group, and a phosphate group. So what I have on the left is the molecule glucose. What I have on the right, that is the product of this reaction, is a molecule that's very, very similar to glucose, except it has a phosphate group on carbon number 6. So here's carbon number 1, 2, 3, 4, 5, 6. So what should I name this molecule? Well, again, it's glucose, except I've added a phosphate group to carbon number 6. So I'm going to call it glucose 6-phosphate. Not because there are 6 phosphate groups, there's one. The 6 indicates it's carbon number 6. Remember back when we did fatty acids, we said that the chemists like to number their carbon atoms. Well, there's carbon 1, there's carbon 6, there's a phosphate group. This is called glucose 6-phosphate. So it's got lots of potential energy still, just like the glucose did. There's still a lot of hydrogen atoms there, still a lot of reducing power. It's still a good fuel, except now it's more reactive than the plain old glucose molecule was, because the phosphate group makes it be more reactive. That's the very first reaction of this process known as glycolysis, which is 10 reactions long. Why am I telling you all this? Well, for a couple of reasons. One, I wanted you to understand that phosphate groups sort of embolden molecules to make them more reactive. That's a handy thing to know in, going forward in biology. But I also told you because I wanted to ask you a question. Where did that phosphate group come from? It didn't just fall out of the sky. The enzyme that picked up the glucose must have also picked up some molecule that could donate a phosphate group, so that phosphate group could be attached here. Well, guess what molecule that was? It was ATP. So the two starting materials of this first reaction of glycolysis are your glucose and a molecule of ATP. Remember, this is happening inside of a cell. So cells have ATP. There's always some ambient ATP laying around. So you pick up an ATP, you rip off one of the 3 phosphates, and you attach that phosphate to carbon number 6. And then you let go of the products. The products are adenosine diphosphate, in other words, ATP minus a phosphate, and this, glucose 6-phosphate. So all you did really was you transferred a phosphate group from ATP to glucose. You start with what's on the left, you end with what's on the right. And you may look at this and say, well, wait a minute, this is backwards. We're doing this process of glycolysis because we want to make ATP. We've done the first step of 10, and we're already in debt by an ATP. We've spent an ATP. We didn't make one. Yeah, that's true. In fact, if you look a couple steps later, you're going to spend another ATP on the third reaction. However, you're going to make ATP later on in the process. In later steps of the process, you're going to make back what you spent and then some. There's no way that you can get the glucose to participate in the series of 10 reactions unless you first sort of jumpstart it by attaching phosphate groups to it. And in the first step, you do that. The only way to power that process, the only way to get the phosphate group and attach it here is to actually spend an ATP. You got to invest something before you're going to get a return. We will get a return. Here's an initial investment of ATP. So that it's okay that the cell does that. It's a necessary step. So I want to, we've highlighted the activity, the idea of making this molecule more reactive, but I want to talk about the enzyme that's involved in this first step as well. The enzyme that catalyzes this reaction is hexokinase. And I'm bringing this up for several reasons. First of all, look at the name. It ends in an ASE, and you know from our previous discussions that enzyme names almost always do, so okay, it's an enzyme. You know that the root hexo, remember back when we did fatty acids and we looked at some Greek roots like tetra and hexo and octa? Hexo means 6. Glucose is a 6-carbon sugar, or what a biochemist sometimes calls a hexose sugar, because there's 6 carbons in it. That's where the hexo comes from here. I'm starting with a 6-carbon sugar. Most importantly, though, the main reason why I'm telling you about hexokinase is this. That term kinase, this molecule is one of many kinases in nature. You're going to see them all the time. If you take other biology classes, you will see that there are lots of different kinases. So let's spend a minute talking about what they are. Kinases, they're enzymes, and they all share a common feature, whether it's hexokinase or any of the other dozens of other kinases that exist. A goal of a kinase is to add a phosphate group to a target molecule. Kinases add phosphate groups to other things. For example, if you take a physiology class, you'll run across a number of different protein kinases. There are enzymes that add phosphate groups to proteins. And there's lots of others as well. So because I know this is a term that you're going to run across in future biology classes, let me just tell you that it's really not complicated. If you are dealing with any kinase at all, whether it's hexokinase or some other kinase, the goal of that enzyme, the purpose of that enzyme, is to add a phosphate group to some other molecule. That's all. That will be a valuable thing to know going forward, so keep that one stashed away for future use. So here we are with hexokinase. This is the first, as I said, of 10 steps in the process known as glycolysis. Again, you can look them up, or I'll post this particular drawing if you'd like to take a closer look at it and investigate each one. But in each step, you're either adding a phosphate group or rearranging some atoms or adding another phosphate group or breaking the thing into pieces or stealing hydrogen atoms, or you're processing the molecule in some way so that you turn one carbohydrate into a different one, and so on and so on and so on. But in a couple of key reactions, for example, right here and right here, you're going to make some of those valuable molecules that we've already talked about, whether it's NADH or FAD or ATP. I'm sorry. So, for example, here's a reaction, and again, you don't have to know the names of these, but here's a reaction where I take this carbohydrate, steal a hydrogen atom from it, give the hydrogen to NAD, and wind up with that molecule. Well, if I give a hydrogen to NAD, I've got NADH. That's a useful molecule. Go back to the previous video and we talked about NADH. So what do we end with? Like I said, I don't want to get too bogged down into the individual reactions, but I do want to say, what do you start with, what do you end with, etc. So back to page 59. We've got this carbohydrate glucose. In a series of reactions, we're going to rip some hydrogens off of it and give them to NAD. In other words, we'll make some of these. We're going to make some ATP directly. There are actually a couple of steps where some chemical bonds are broken that release so much energy that the cell is able to attach ADP to a phosphate group. And when you attach ADP to a phosphate group, you get ATP. So there's enough energy stored in this original glucose to power the production of some ATP directly. We'll make some of them. But there's got to be something left over at the end of all this. If I start out with the carbohydrate known as glucose and turn it into this and then to this and then and so on and so on and so on, what's the carbohydrate that I'm left with? I'm left with a carbohydrate called pyruvate. That's the molecule pyruvate right there. It's a really important molecule in biochemistry. You'll see it over and over again in biology classes. And for our purposes today, I just want to note that it is the ending material of glycolysis. So let's summarize the process of glycolysis. We went into the detail on the first reaction, that's fine, but I really just want to jump to the end now and say, what did we do? What did we get out of all this? Well, for every one glucose molecule that a cell starts with in the process of glycolysis, by the time all is said and done, you will have produced a net of 2 ATP molecules. You will have produced 2 molecules of NADH, and the leftovers, the carbohydrates that you have laying around by the end of all this, are those pyruvate molecules, and you'll have 2 of them. Notice that glucose has 6 carbons, pyruvates have 3 carbons each, so it makes sense if I take this and process it and chop it in half, I'm going to have 2 of these for every one of those. So here's the take-home message. When a cell starts with glucose and runs it through those 10 reactions of glycolysis, it does some good stuff in that it makes a couple ATP. It does some more good stuff in that it makes a couple molecules of NADH. And the leftovers are these pyruvate molecules. Now, pyruvate molecules are still pretty reduced. They're still pretty valuable. They're not as energy rich as glucose was, but they still got some potential energy in them. If all you do with your glucose is glycolysis, you're not getting the most out of it. You're not being very efficient. You're not squeezing as much energy as you possibly could out of that original molecule. Cells like ours and other animal cells can't rely only on glycolysis, which is why we've got phase 2 and phase 3 that are coming up. But to draw the line for the end of this video, I want to just focus on glycolysis. Your starting material is glucose. Your ending materials are 2 ATPs, 2 NADHs, 2 pyruvate molecules for every one glucose that you start with. Now, just to wrap up a couple more things before we end this video and talk about Krebs cycle in the next video. Where does all this happen? I said it happens inside of a cell. That's fine. But is there an organelle in which glycolysis happens? No. Glycolysis happens just in the cytoplasm. There's no special organelle. It doesn't happen in the mitochondria or the chloroplast or any place else. It happens just in the cytoplasm. Why? Because the enzymes that catalyze the reactions of glycolysis are in the cytoplasm. So that's where the reactions happen. Something else to sum up here. At the top of this page, I said, how is the energy extracted from glucose during glycolysis stored? Well, if you go back to page 54 of this book, you know we listed different ways that cells deal with or reckon energy. We said there are chemical bonds, for example, the bonds between the phosphates of an ATP. We said there's reducing power, richness in hydrogen atoms, that is. And we listed a third there that we haven't talked about yet. But think about what we made. We made ATP. Why is ATP valuable? Because of those high energy bonds between the phosphate groups. So chemical bonds, specifically in the ATP molecule. We also made some reduced coenzymes, some reduced NAD, otherwise known as NADH. So we transferred reducing power, or hydrogens, from a carbohydrate to a coenzyme. Now the coenzyme is holding reducing power. Look back on page 54. Reducing power was one way that a cell handles energy. There's an example right there. So ATP is the example of this, NADH is the example of that, right here. And then, as I said, we're also left with this pyruvate molecule, with a couple of them actually, that are pretty valuable, just not as valuable as the original glucose was. We don't want that to go to waste. We want to continue to metabolize this thing and extract even more energy from it so we can make even more ATP. But that's a separate process. Glycolysis ends right now. We can take that pyruvate that we make in glycolysis and run it through another series of reactions and squeeze even more energy out of it. That's what this process called the Krebs cycle is going to be. It's another series of chemical reactions. Because when you consider this, this doesn't look all that impressive. At the beginning of this discussion, we said, oh, yeah, glucose is great. It's loaded with potential energy. Look at all that reducing power. It's fantastic. Tons and tons of potential energy. And all we got out of it was 2 lousy ATPs. That doesn't seem very impressive. Well, it's because you're done with glycolysis, but you're not done. You're not done with carbohydrate metabolism. This is just phase 1. We're going to get more efficient coming up. So please go back and review that. Think deeply about what you just did to this carbohydrate known as glucose. Think about what you ended with, and the fact that what you ended with, pyruvate, is still very valuable. There's still hydrogens attached there that I could rip off of there and oxidize this further and say, oh, look at that. I can do even more work. This is great. But I got to do a different set of chemical reactions, and those are the ones coming up known as the Krebs cycle. I'll put those in a separate video.